Hi guys, I think we're ready for another whiteboard talk. We're going to take a look at meiosis and look at all of the details of the meiosis 1 and meiosis 2 division. Remember, in meiosis, one cell is going to give rise to 4 genetically different cells that are haploid. And so in my drawing, I've got a cell up here, and this cell is preparing for meiosis, and this cell, it says 2n equals 4. Remember that means that it's a diploid cell, and its diploid number is 4. It has 4 total chromosomes, or 2 sets of chromosomes, 2 sets of 2 chromosomes, all right? And in this, it's an animal cell, and there is its genetic material. Probably it has nucleoli in its cell. This cell is a cell that's going to give rise to the sex cells, or the gametes, right? And down there is a centrosome. Before this cell can go through meiosis, this cell has to go through a pre-meiotic interphase in which it's preparing for meiosis. And one of the important things that happens in the pre-meiotic interphase during synthesis is its DNA replicates, so that each string of chromatin is genetically replicated so that there are 2 copies of all of the genetic material. And also, the centrosome is replicated. Remember, that's the microtubule organizing center of the cell that's going to be important in creating a spindle later. There's our cell. Now that cell is ready to go. It's got 4 strings of chromatin right now that have all been replicated, and it's ready to go through meiosis. When it gets done, we should see the making of eggs or sperm that have a haploid number of chromosomes, right? That have n equal to 2. We'll see how that goes. Now, it turns out that meiosis is a 2-phase, there are 2 divisions in meiosis. Meiosis 1, and we'll start with meiosis 1, happens first, obviously, and in meiosis 1, we call meiosis 1 the reduction division, because in meiosis 1, the chromosome number is cut in half. It's reduced. So you can put that in your notes that this is the reduction division, and we'll see that as we go, right? When we talk about meiosis 1 and meiosis 2, we divide up meiosis into phases, just like we did with mitosis. This time we're going to divide meiosis 1 and meiosis 2 into phases, into 4 phases each. Prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2. We're not focusing on prometaphase with our meiosis, all right? And so this cell is now ready to go through meiosis. It has replicated its DNA, it's replicated its centrosomes, it's looked at itself, and it is ready to divide. So it starts meiosis 1. And meiosis 1, the first step would be called prophase 1. What happens in prophase 1? And I've got my notes with me to make sure that I catch all my details, right? This is a very long phase. First of all, in human females, this phase can last upward of 50 years. That's crazy, right? Well, what happens in girls who aren't even born yet is some of their cells in their ovaries start to go through meiosis, and they stop in prophase 1. There might be 400,000 of these cells that have started meiosis, started prophase 1, and then just stop. And then once a girl goes through puberty, about once a month, one of these cells continues through meiosis to become a mature egg so that it could be fertilized. And that can happen month after month, year after year, until a woman is close to 50. So this is a really long phase, at least in humans, in human females, all right? All right, what are some of the things that happen? Let's write some things down. First of all, the chromatin condenses. The chromatin condenses to form chromosomes. So now we're going to be able to see our chromosomes. Each chromosome should be replicated, so every chromosome is going to look like this. Each chromosome is made up of 2 sister chromatids that are genetically identical, okay? Things like the nucleoli disappear. Late in prophase 1, the nuclear envelope ruptures. So I'm just writing Ne for nuclear envelope there, all right? And the spindle starts to form. We're not going to call it the mitotic spindle this time. It's meiotic spindle, but the spindle starts to form. And that's a network of microtubule fibers extending between the two centrosomes. I haven't drawn anything in yet. I will start to draw in our spindle a little bit, okay? So there's our spindle. It's starting to form and starting to grow. And then really importantly here, this is different than mitosis. Homologous chromosomes pair up or come together in a process called synapsis. So I'm going to write that down. Homologous chromosomes pair up in a process known as synapsis. Let's draw this out a second. And let's think about this with our little pipe cleaner chromosomes. I know I'm going in and out of the picture a little bit, but that's okay. All right, so what we've got in our cell should be 4 replicated chromosomes. 2 of them are long and 2 are short. Got these guys down here. I can use my pipe cleaners to identify these, okay? So I have 4 replicated chromosomes. Oh, my replications aren't sticking together here. My chromatids aren't real happy. Okay, this guy looking a little weird. Now what happens in prophase 1 of meiosis 1 is homologous chromosomes. Homologous chromosomes, remember, are those that have the same size, the same shape, they carry genes for the same traits. Homologous chromosomes pair up. They come together. Okay? There's a complex of proteins called the synaptoneme complex that helps to zip these homologous chromosomes, kind of pair them up. And when they're paired up, we say that we sometimes call them a tetrad. Can you see why? There are 4 chromatids, right, in this tetrad this is a set of homologous chromosomes that have paired up in the process of synapsis. All right. This never happens in mitosis. So this is kind of cool and different. It's going to be really important. All right, so once they pair up, you've got homologous chromosomes, and we call their chromatids non-sister chromatids. Non-sister chromatids would be like the green and the orange ones. Sometimes kind of cross over each other and form crosses or chiasmata is the singular. And when these crosses form, we call them, we actually call them crosses or chiasma. Sometimes genetic material is actually swapped between the chromosomes. So a little bit of the orange chromosome ends up on the green chromosome. A little bit of the green chromosome ends up on the orange one. What that leads to is genetic variability in our chromatids. They're not all genetically identical anymore. The 2 green ones aren't identical because the green one gets a little bit of orange. The 2 orange ones aren't identical because the orange one gets a little bit of green. This is one source of genetic variation that shows up in prophase 1 of meiosis. And it's called crossing over or genetic recombination as these sister or non-sister chromatids swap genetic material. And it starts in prophase 1. It takes a little longer to complete the process. But I'm going to draw that out, and then we're going to draw out our chromosomes as well, all right? So let's draw out, so we could have, a pair of chromosomes, pair of homologous chromosomes that have lined up. They're close to each other because of that synaptoneme complex kind of holding them together. I can draw that in too if you want, this synaptoneme complex of proteins. And then what happens is crosses form between these. So they get tangled up with each other, and a chiasma forms, or a cross forms in between or connecting or crossing the non-sister chromatids. Oops, I drew in the wrong color there, okay? Lots of those crosses can form. eventually that leads to the swapping of genetic material or genetic recombination. That's huge. That's a source of variability in meiosis. And so I'm going to draw in my 4 chromosomes because they should all be visible. The nuclear envelope is already ruptured, so I guess I'm going to have to rupture my nuclear envelope. I'm looking for my markers. I'm always losing them. I've already lost a black one. But I'm going to draw in a ruptured nuclear envelope, and inside there I'm going to draw my chromosomes. Remember, they're paired up. The homologous chromosomes are synapsed, so I'm going to draw in one long red chromosome, one short red chromosome, one long green chromosome, and one short green chromosome. And then I might want to draw in some crossing over that's happening. So I'm going to draw in, I might make some X's in my drawings. Oof, this is hard to, oh, whoops, I did it wrong. Kind of constantly doing this wrong here. I'm going to draw an example of a chiasmata forming there, a cross forming, and that's where genetic material is going to be swapped at some point during this process. Okay, this was a big step because we've never seen homologous chromosomes pair up before, not in mitosis. This only happens in meiosis, in prophase 1 of meiosis 1. All right, now we're moving on. What happens next? After prophase 1 comes metaphase 1. Right? In metaphase 1, let's draw it out. Okay, we've got a circular drawing there. All right, in metaphase 1, the spindle, the spindle has now entered the nuclear area. Okay, so let's write that down. Spindle enters nuclear area and attaches to the chromosomes. But it attaches in a very particular way, and we should draw that out somewhere. So let's draw that out over here. Let's say I've got my pair of long chromosomes, my homologous pair of chromosomes, and there's probably some crossing over happening, but I'm not going to draw that in. What happens with a spindle, this spindle fibers attach to one of the homologs, both sister chromatids of it, from one side of the pole, and spindle fibers from the other pole attach to the other the other homolog, the other homologous chromosomes. Because what's going to happen to these chromosomes eventually is the homologous chromosomes are going to separate from each other, right? And then there's a little bit of a tug-of-war that goes on. This pair of homologous chromosomes are kind of getting pulled and pushed up and down in this cell and end up kind of in the middle, at the metaphase plate. So I'm going to write that homologous chromosomes align at the midline or at the metaphase plate. Now, how do they line up here? Well, let's draw them in. Let's draw in an example. So I have these 2 pair of chromosomes. Okay? One long green, one short green, one long red, kind of now in the midline, and one short red, paired up with its homologous partners, right? So our homologous chromosomes are paired. The spindle fibers are attached properly, we hope, so that homologous chromosomes are attached to fibers from opposite poles. Okay? And, then there's one more question that I have to ask, and that is, how do these pairs of chromosomes line up at the midline? Do they line up so that, like, all the green chromosomes are always on the top and the red chromosomes always on the bottom? Or does the way that this pair of homologous chromosomes line up at the midline, is it independent of this one? The way that chromosome pairs line up at the metaphase plate is independent of each other. So in my drawing, the long green chromosome ended up on the top part of the cell. The short green chromosome ended up on the bottom. It could well be that the short green chromosome also ended up on the top part of the cell, okay? And the short red chromosome down here. They're still paired together. They're not real close there. They should be a little closer. The way that one pair of homologous chromosomes lines up at the midline is independent of the way that the next pair lines up. And if we have 23 pair of chromosomes, there is the independent assortment of chromosomes for all 23 pair. What does that mean? It means that as this meiosis happens, you're going to end up with different combinations of chromosomes in opposite cells. In this drawing, both of my green chromosomes are going to end up in one cell and both of my red chromosomes in another. But if I redraw this with my short green chromosome on the kind of the bottom cell, the bottom forming cell, and my short red chromosome